Here we have the DNA structure with matching nitrogenous bases. This represents the 5 to 3 prime and how they are anti-parallel. This part represents the hydrogen bonds, where there is 2 hydrogen bonds in between the A and the T, and there are 3 hydrogen bonds in between the G and the C. Purines have 2 rings and pyrimidines have 1 ring. Pyrimidines are C and T, and primines are A and C.
Semi-conservative replication DNA double helix unzips, and each strand serves as a template for the formation of a new strand. The new molecules each consist of 1 old strand and 1 new strand. DNA separate to form replication bubbles, which expand to replication proceeding in both directions at once.
The major steps of replication are first, helicase, which unwinds the DNA at origins of replication. The second, initiation of proteins separate 2 strands and form a replication bubble. The word that Allison is writing out that starts with a T relieves overwinding strain ahead of replication forks by breaking, swiveling, and rejoining DNA strands. The single-strand binding proteins binds to and stabilizes single-stranded DNA until it can be used as a template. The primase puts down RNA primer to start replication.
DNA polymerase replicates the 2 original strands of DNA differently, although it binds both new strands from the 5' to 3' direction. 1 strand is formed towards the replication fork in an unbroken linear fashion. This is called the leading strand. The other strand is the lagging strand, in which it forms in the direction away from the replication fork, in a series of segments called Okazaki fragments.
Here is shown DNA polymerase 1, which removes RNA nucleotides of primer from 5' and replaces them with DNA nucleotides. This is the DNA polymerase 1, which replaces the RNA primer with DNA nucleotides. This shows the leading and lagging strands. This is the 5' and 3' of the old DNA, and this is the 3' and 5' of the new DNA. What Allison is circling are the primers that start off the strands.
Okazaki fragments are about 100 to 200 nucleotides long and will be joined into 1 continuous strand by the enzyme DNA ligase.
The energy that creates the growing strand comes from the incoming nucleotides. The reason why the replication on the lagging strand is discontinuous is because the other template strand is oriented 5' to 3' at the replication fork, and hence copying it will result in the synthesis in a 3' to 5' direction relative to the direction of fork movement.